In this lecture, we will talk about the methods of creating new variation in case that your germplasm or collection lacks certain level of genetic variability for the trait or gene of interest. So here's the outline. So we only have two topics. So we will talk about the naturally existing variability and the methods to create new variation. So here is a schematic diagram of most plant breeding programs, which starts with a breeding goal and then followed by creation of variation and population formation, selection, evaluation, and the creation, certification and cultivar release. So for this lecture, we will focus on the creation of variation, and there are two ways on how to increase your genetic variability. You can use the natural existing variability, or you can do a method to create new variability. So let's start discussing the naturally existing variability, the domestication, germplasm collection, and plant introduction. I have already introduced you the term domestication, which is the collection of wild species and putting it under human care. So you might think that domestication occurred already in the past, but it's not true because it's an ongoing process. In fact, 230 plant species have already been domesticated since then. Germplasm collection is also another source of naturally existing variability. Germplasm is the total sum of hereditary materials, and your sources of germplasm materials are the centers of origin, diversity, gene banks, sanctuaries, farmers' field markets, and even seed companies. So germplasm collection is a valuable resource of plant genetic diversity because you can find many different types of genetic material, say for example domesticated or the domesticates wild relatives, indigenous and exotic materials, land races, modern and obsolete cultivars, and even breeding stocks. So those are the different materials you can find or collect from germplasm collections. Plant introduction is another way in order for you to increase the genetic variability, and it's natural because you don't need to do something. You just have to import or introduce. So plant introduction is actually the movement of germplasm from one area of the world to another. Or simply, let's say, for example, if you collected varieties or genotypes from other countries, say for example Japan, and then in Japan you collected different types of or different genotypes of taro and you introduce here in the Philippines, by that you're doing plant introduction. So again, here are the naturally existing variability wherein you can collect germplasm to increase the genetic variability of your collection or your own germplasm. So domestication, germplasm collection, and plant introduction. So we have already discussed the three naturally existing variability, and we will now discuss the methods to create new variability in case you don't have access, for example, in the germplasm collection, or you don't have any materials to introduce. Then you can go to these five methods: hybridization, mutagenesis, polyploidization, somaclonal, and the last is genetic engineering. So plant hybridization is defined as the crossbreeding between individuals with contrasting traits. So here the key word is contrasting traits, because if you hybridize or you cross individuals that are or have similar genetic content or genetic makeup, then the chances of producing new variability is very low compared to crossing uh, totally different or parents that have contrasting traits or with different genetic makeup. So there are two ways on how to hybridize. You can do intraspecific hybridization. So from the word intra, so it's within. So here within species. So if you cross two genotypes of the same species of corn, then you are doing intraspecific hybridization. On the other hand, if you cross two organisms or individuals with or coming from different species, like for example that the Yosente and our modern corn, then that's interspecific hybridization. Okay, so these two types of hybridization has been used by breeders in order to create new variation, or to introduce new genes coming from the wild species just to increase the genetic variability of their material. So again, if you do hybridization, always remember that the two parents or the genotypes that you're going to cross must have contrasting traits. So say for example, we have here capital A's, small letter B's and C's, and capital D. Those are, let's say, the loci of the genes, simply the genes of this particular individual. The other individual has a contrasting trait, say for example, small letter A, capital letter B, capital letter C, and small letter D. And as you can see, if you're going to compare them, they have contrasting genes. So if you cross them, you will produce this what we call hybrid or F1 generation. But here, you cannot have genetic variation because all of the offsprings between these two parents will produce a homogeneous but heterozygote individual. And so all of them, they have the same genetic makeup. Only then you can have new variation if you sieve or you self this F1 individual, and after you self them, there will be now segregation of alleles and genes, so you will produce different kinds of plants having different genetic makeup. So here you have capital A's heterozygote for BZ, B loci, and then small letter C, and heterozygote for D, so on and so forth. So that's hybridization. By the way, this is intraspecific hybridization. You can also do intraspecific hybridization, but you'll be facing with some difficulties because you will have a linkage drag. So what is linkage drag? It's when you do hybridization, instead of having the desired trait, you will also obtain undesirable trait, which is that might be deleterious for the individuals or the hybrid. The second method to create new variation is through mutagenesis. We have already discussed what mutation is and the types of mutagens used to induce mutation. So mutagenesis will only be effective if the mutation occurred in the protein coding region of the DNA. So remember, DNA, in the expression of genes, there will be splicing. So if mutation occurred in the non-protein coding region, then there will be no observable effect on the phenotype. And all mutations except silent mutations should create new variability. So for example, indels, so insertion deletions, and substitution will create new alleles, and that allele will you express will show another phenotype, and that's a mutant phenotype. So for example, we have here gene DTH9, which when they mutated, it regulates disease susceptibility in Arabidopsis. Mutagenesis has already been used by plant breeders in different crops or different species to produce new varieties. And in fact, around almost 50% of cereal varieties were produced by mutagenesis. All the others, let's say for example, for flowers or ornamentals, and legumes and other types of crops. Polyploidization is another method to create new variation in plants. So polyploidization is actually the technique of multiplying the chromosome complements in a cell. The chemicals used to induce mutation can also be used to induce chromosome doubling, such as oryzalin, trifluralin, and colchicine. In fact, this colchicine has been used to improve the quality traits in many ornamental species. Say for example, glandulus. So the effectiveness of colchicine in chromosome doubling or to induce polyploidization depends upon the concentration of the chemical. So here in our example, we have here the normal or the control glandulus, and letter B is the concentration here is 0.1%, and as you notice, there's not much increase in the size. In fact, it has a lower. If you are going to see it by your eyes, the flower is smaller compared to the control. But by increasing the concentration of colchicine, the researchers observed that Some of the plants flowers or the florets has increased. Here you have a bigger floret that's in C and that to be compared to the control. So how will you check if the chemicals you used for chromosome doubling was effective? So you can use flow cytometry. So the principle in flow cytometry is the use of laser beams to emit or to produce fluorescence intensity and discriminate the sizes of the chromosomes or the cells that you load in the equipment. So here we have first our suspension. So here an example is chromosome, or you can also load cell here, and you put it in the flow chamber, and in this flow chamber the chromosomes will interact individually with the laser beam, and but before you load your chromosomes, you label it with dye, and once the laser hits the individual chromosome, and it will fluoresce and emit fluorescence intensity, and it will be sent to the computer or your desktop and shows you the data. And here farther down, once this laser interacts with the individual chromosome, if say for example some of the chromosomes are smaller, so they will group them based on the size. So if you have cells, if you have diploid, tetraploid, hexaploid, then they will group them after the laser hits each of those cells. So if you, in case you use cell, you will have this kind of data. So you will have here the number of cells, and here you have the different types of ploidy level. So you have here the chamber. So this flow karyotype may also look like this, but this one is for the sizes of the chromosome. But here on the right are the sizes of the cell that you load in the instrument. So that's how you use flow cytometry to detect if the cells that you expose to the different concentration of colchicine has doubled the chromosome, or tripled, or even quadrupled times. Somaclonal variation is another useful technique to create new variation. This is somaclonal variation is actually the variation observed among cultured plant tissues. So what you do here is that first your tissues or your cells, you put them in a medium and allow them to produce callous tissues, and then later with certain time you induce that callus to produce organs. So that process is called organogenesis. And then here you regenerate the plants, and once they grow you will observe these somaclonal variants. So when or how does these variants are produced? So during culture or plant cell and tissue culture, certain in vitro conditions causes chromosomal rearrangement or epigenetic changes. So basically this occurs if you elongate or you prolong the callus phase of the cells before you induce them to produce organs, the roots and the shoots. So to develop crops tolerant to abiotic stress or resistant to biotic stress, you can use somaclonal variation. And once you have the plantlet, you can expose them either in a drought for abiotic stress, or you expose them, or you infest certain disease or certain insect. And then, of course, if there are lines that are resistant or tolerant, then you can select them. So the main goal of somaclonal variation basically is to produce or to create new variation for breeders to select on. So as you can see here. The last method is through the use of genetic engineering. So in some other countries, this genetic engineering is commonly used and is allowed, unlike here in our country. We will be facing some difficulties if we do this genetic engineering, but maybe probably if your reason is for educational purposes and not for commercialization, then maybe you can secure some biosafety uh, rules or biosafety agreements in order for you for the authorities to allow you to conduct genetic engineering. So it's a modern approach using biotechniques especially the use of recombinant DNA technology. I have shown this picture before. It's a diagram used in the recombinant DNA technology, when you have here plasmid and you have your linearized DNA. And what you do with the plasmid is you also linearize, and with the use of endonucleases, you linearize that, and with the use of some ligase, you allow this fragment, linearized fragment, probably this will be your gene of interest, and you insert that on the plasmid and allow it to connect and then form a circular bigger plasmid. And of course you will do transformation. You transform first your E. coli, and then once you propagated your E. coli, of course you also propagated your plasmid, your vector, and then you will extract, or you will harvest those plasmids, and you will transform that again to agrobacterium and infest your agrobacterium to your crop in order to deliver the gene of interest inside your the cell of your crop of interest. So again, you can manipulate the gene using recombinant technology, and the ultimate goal is to insert a parent DNA into the host cell of your crop.